Hi everyone, good morning. Hope you are doing fine, and we will start the next lecture, which is Antimicrobial Resistance Part 1. So what we are going to discuss today is mode of action of different antibiotics, application of antibiotics for bacterial pathogens, what is AMR or antimicrobial resistance, and what is One Health and why does it matter. So you might already know some of the answers to these questions, but we will discuss in a step-by-step manner. So yesterday we all saw the distinction between antimicrobial and antibiotic, right? What is the difference between the two? Quickly, someone wants to. Different types of microbes, yes. So because as the term is suggesting, antimicrobial. So antimicrobial agents will also include antibacterial agents as well in it, remember that. But in antibiotic, you have got bacteria only that are targeted. So those are mostly antibacterial agents. You know, while saying, you will not say, I want an antibacterial agent, right? You have a very general term, which is antibiotic. Give me an antibiotic, okay? So antibiotic refers to substances produced by microorganisms that can act against other microbes, and these are all mostly antibacterial agents. And antimicrobials, as you said, are against different sorts of microorganisms. So antimicrobials are classified, based on their importance, and WHO or World Health Organization ranks antimicrobials based on the importance of human medicine. Antimicrobials have to fulfill specific criteria and factors to be deemed, to be deemed as highest priority. For example, critically important antibiotics or important antibiotics. There are different categories over there. And on the right hand side, again, you see a picture or a snapshot of the document listed by WHO that will highlight, different areas of priorities that are there. So for example, penicillins are such broad class that they are split into 6 groups. And yesterday itself you saw that there are different antibiotics under the beta-lactam category, for example. So this is just an example highlighting CIA and HIA. So CIA is critically important antibiotic, HIA is highly important antibiotic or antimicrobials, and you can see the examples of these. What you need to appreciate is that in the critically important antibiotics and in highly important antibiotics, you can have the same class as well. You can have the same class that is there, but the antibiotics that are there in the category can be different, So for example, in the carbapenems, you have imipenem, meropenem, doripenem, ertapenem. So they can be split based on how they are classified. So very nicely, you can already see, for example, in the critically important antimicrobials, you can see in aminoglycosides, you have got, for example, gentamicin. Then vancomycins, you have got rifampicin, which is used for TB, for example. Then in carbapenems, you have got like meropenem, and so on. There's a list of things over there. In polymyxins, you have got colistin, which is another important antibiotic. And in highly important antimicrobials, again, you have got, amphenicols like chloramphenicol. In cephalosporins, you have got first and second generation, for example, cefazolin and, other cefamycins. In lincosamides, you have got clindamycin and so on. So you can see. So in penicillins, for example, you can see like flucoxacillin is there as a highly important antimicrobial over here. On the other hand side, for example, in the critically important one, you have got ampicillin, piperacillin that are critically important, and that is what I meant. You can have penicillin in the CI, you can have penicillins in the HI as well, but then the specific antibiotics that are there, um, you know, whether it is ampicillin, piperacillin, or flucoxacillin, or anything else, can be in different categories. So we discussed this yesterday as well. So this is just a quick recap highlighting, the different classes are there. You have got the beta-lactams, where you have got penicillin, cephalosporin, monobactams, carbapenems. So these target both gram-positive and gram-negatives, and examples have been given. Penicillin G, ampicillin, piperacillin, and so on. In cephalosporins also you will have several generations of cephalosporins that are there. As I told you, as the generation increases, the antibiotic becomes more stronger, basically. In monobactams you have aztreonam, and carbapenems you have got imipenem, meropenem, and mode of action of this is to inhibit the cell wall synthesis. In aminoglycosides, you have got gentamicin, streptomycin, tobramycin, amikacin, neomycin. Many are there. They mostly target gram-negative organisms, inhibit protein synthesis in 30S subunit of the ribosome, bacterial ribosome. In tetracyclines, they are active against positives as well as negatives. And examples of tetracycline antibiotic is tetracycline itself. And doxycycline is also there, again inhibition of protein synthesis, 30S subunit. In macrolides, you have got erythromycin, for example. Again, it targets gram positives. Here, mode of action is inhibition of 50S subunit. In glycopeptides, target gram positives. And examples of these are vancomycin, teicoplanin, and they again inhibit cell wall synthesis. And then sulfonamides will act against positive and negative. An example of that is sulfamethoxazole, and that inhibits, for example, folate synthesis. And then lastly, fluoroquinolones that target gram positive as well as negative. An example of that is, getifloxacin, ciprofloxacin, norfloxacin, levofloxacin. So many are there. The nalidixic acid is there. So mode of action is inhibition of DNA replication over there. So we will start with the first one, penicillin, right? Everyone, the whole world knows penicillin, right? Is the first what? What is first? Antibiotic or antibacterial agent, anything. Okay, so this is bactericidal, okay? When I say bactericidal, what does it mean? Kills completely, okay? So it is highlighted there. Penicillin kills bacteria through binding of the beta-lactam ring to DD transpeptidase, which is a penicillin binding protein. Excuse me. Now you can see very nicely in that particular chart or the diagram flow chart on the right hand side, you have got the bacterial cell over there, and the cross links are supposed to be formed, and you have got the penicillin binding protein. So penicillin binding protein is supposed to go there, and it is supposed to help in developing these cross links. But what happens is that penicillin, the antibiotic that is there, it binds to that penicillin binding protein. And when that penicillin binds to that penicillin binding protein, the reaction that is supposed to catalyze, in order to build the bridge between the different residues, for example, in the reaction that is supposed to happen in 4 and 5, step number 4 and 5, that reaction can't happen, so that bonds are not formed. So when those bonds are not formed and those links are not developed, obviously the cell wall is weak. So bacteria can't survive as a result of that. I gave you the example yesterday. I told you the wall. The wall is like, it is nothing. It is same as this wall, and bacterial cell wall is the same analogy. You know, you have the bricks over here, you have got the N-acetyl muramic acid and acetyl glucosamine that are there, and the links are being formed. You know, beta 1,4 glycosidic linkages that occur. Um, and if you have a boulder to hit that wall, then that is the penicillin basically, you know, which can break it. But here you can see that, you know, this is what is happening, you know, when the penicillin binds to that protein. So it inhibits the cross-linking activity, as I highlighted, preventing new cell membrane formation. Without this cell membrane or cell wall, the bacterial cell is vulnerable to outside water, because we all know in a construction you need a very solid construction. If you don't have a solid construction, you know what is going to happen to the building. What? It is going to collapse without doubt. So the same thing happens over here. When the cell is vulnerable to the water molecules, and you know the pressures and all are, you know, different inside and outside the cell, it will lead to the death of the cell. Okay, it is very clear. And then in the last point, since human cells don't contain cell wall, penicillin treatment results in bacterial cell death without affecting the human cells. Okay, that is what, and that is another reason that I highlighted earlier. You know, how antibiotics are selective against the organisms it themselves, and uh, uh, not harming the human cells, for example. Then the next one that we have is gentamicin, right? So gentamicin is a bactericidal antibiotic in a higher concentration. Mostly gentamicin is bactericidal. Gentamicin belongs to which class of antibiotics? Gentamicin is in which class? Aminoglycosides. Okay. So there are certain things that you need to remember. I mean, I know, I appreciate there are students here from the non-biology background or whatever, but there are certain things that you need to know, because that basic foundation, without that you will not know anything else. So you need to know that, okay, these classes are there, and then in these classes these antibiotics are categorized. You know, there is no other way of remembering, or it is like learning the letters of the alphabet. So there you will have to know few things. So gentamicin is aminoglycosides, right? So it is bactericidal. It binds to the 30S ribosomal subunit and disrupts the process of protein synthesis by preventing the amino acid tRNA access to the A site on mRNA ribosome complex. It also leads to distortion of the 30S subunit. So when the 30S subunit is distorted, that time also that protein synthesis that is going to occur, the initiation of protein synthesis that is supposed to happen will not happen, because the codons can't align to the tRNA. Uh, and this binding over here, this is an irreversible binding. It is not highlighted over here, but this binding is an irreversible binding. You know, in reactions, there are reactions, some reactions are reversible, some reactions are not reversible. So over here is irreversible binding. So it blocks initiation of protein synthesis and blocks further translation, resulting in cell death. So some aminoglycosides will interfere with proofreading process that helps assure accuracy of translation. So as a result of all these issues, the uh, the activity will be seen and the cell will die as a result of that. So tetracycline is a bacteriostatic antibiotic, and this also works by attaching to the same like 30S subunit of the ribosome, binds to 30S ribosomal subunit and prevents the attachment of amino acyl tRNA to the A site of the ribosome mRNA complex. So this inhibition blocks the elongation phase of translation. So you can see in the diagram, you have the amino acyl tRNA that is coming, and then the polypeptide chain is forming, and you can see over there tetracycline will sit and it will inhibit that process, so that elongation is blocked. So when the polypeptide chain, you know what is elongation, right? You elongate. So the chain is supposed to grow because this peptide chain is formed and proteins are made as a result of that, and that's how you have the proteins that are formed for cellular processes and other things. So the inhibition blocks the elongation phase of translation, halting protein production, suppressing bacterial growth. But remember that this is a bacteriostatic antibiotic, and this action over here, it is reversible. It is not written over here, but this action is reversible. Then we have got macrolides, right? So macrolide is bacteriostatic uh, antibiotic, and macrolides bind to 50S ribosomal subunit. Okay, specifically to the 23S rRNA near the peptidyl transferase center and the nascent peptide exit tunnel. So this results in inhibition of bacterial protein biosynthesis. As a result, um, in slow growth or proliferation of cells. So as opposed to the previous two where you saw, aminoglycosides, you saw tetracycline, they were mostly, affecting the 30S. Here it is 50S that is being affected. And don't get too confused, 50S and 30S, these are the subunits. These are ribosomal subunits, especially for people who don't know. The bacterial ribosome is made up of 50S and 30S subunit, and that whole thing is called as a 70S ribosome. It is not mathematics over here. It is not 50 plus 30 is equal to 80. Oh, okay. So S is known as the Swedberg unit. Okay, and if people over here already know what that is, it is the rate of sedimentation of ribosomes when you are, centrifuge, when you're putting them in the centrifuge and you are sedimenting them, then that is that level at which the sediment of those units are seen. So that is how the thing is derived. So don't be too confused. So then next is glycopeptides. Glycopeptides are bactericidal. Again, you know what is bactericidal already. So glycopeptides work by inhibiting bacterial cell wall synthesis by binding to D-alanine terminus of peptidoglycan precursors. It physically prevents both transglycosylase and transpeptidase enzymes from cross-linking the cell wall. And you can see that in the diagram. In the diagram, you can see the purple thing, which is vancomycin, and vancomycin will bind over there. And when it binds over there, it will not be able to form the links with each other. The pentapeptide that is seen, and then the pentaglycine bridge that is formed, for example. And I think you know what is GLNAC and MURNAC. It is N-acetyl glucosamine and N-acetyl muramic acid, which is a disaccharide. So transpeptidase is the key enzyme for this process. It cannot function correctly because vancomycin occupies its necessary binding site in the cell wall building block. So it is like vancomycin is sitting over there, not allowing that reaction to occur. So this is again um, bactericidal antibiotic. Then next, talking about sulfonamides. So these are bacteriostatic. Um, so sulfamethoxazole is a very common example of a sulfonamide. Sulfanilamide sulfanilamide is a structural analog of para-aminobenzoic acid, which competes. It competes with PABA, right? So it binds to dihydroteroid synthetase and inhibits conversion of PABA and dihydroteroid diphosphate to dihydrofolic acid or dihydrofolate. So there is a competitive inhibition that occurs. So dihydroteroid synthetase is an important enzyme, and you know, as I said, it binds to dihydroteroid synthetase and inhibits conversion of para-aminobenzoic acid and dihydroteroid to dihydrofolic acid. And dihydrofolic acid is very important. Dihydrofolic acid then goes on to form tetrahydrofolic acid with the enzyme dihydrofolate reductase, and that tetrahydrofolic acid is converted to purines, not just purines but purines and pyrimidines as well. And if you know what's purines and pyrimidines, they are the adenine, thymine, guanine, cytosine, the building blocks of your DNA. So folate is also essential metabolite for bacterial growth, right? So that is another, I told you yesterday, bacteria are producing so many things. So folic acid, another thing, you can get folic acid from outside, and especially in women, right? Women who are pregnant, you will, maybe you can relate in your family, for example, they are prescribed folic acid. Why? For the development of fetus. Okay, that is the reason. It is not just like, you take folic acid because you don't think, no. But now you know where that folic acid is coming and how it is important and what it is actually doing. So it is essential for that, for the growth. So folate is an essential metabolite for bacterial growth replication because it uses DNA synthesis, and blockage of folate production inhibits the folic dependent metabolic processes for bacterial growth. So then we have got ciprofloxacin, which is from the fluoroquinolone category. You have the quinolones, and then you have got the fluorine attached to the quinolones that converts the quinolones to fluoroquinolones. They are more powerful than quinolones. Okay, so there is a distinction between quinolones and fluoroquinolones. So what ciprofloxacin is doing is it interferes with DNA replication. Everyone knows what's DNA replication, right? The DNA is going to open itself, and then Okazaki fragments, I'm sure you know little bit, are synthesized and you have got the DNA. So DNA gyrase is the enzyme, okay, or topoisomerase 2. So that is the enzyme that helps in opening that DNA. It helps in opening. And what happens if you have, if you have a thread, everyone has worked, everyone has sometimes played with a thread sometimes in their life, right? When two threads are intertwined and you pull them apart, what happens at one end? There is stress created. Same thing happens here also. There is stress created over there. So the job of that DNA gyrase is to relieve that stress, okay, to make sure, because if too much stress is created, if you pull, what's going to break? Yes, same thing will happen over here. Okay, so that is the job of the this thing. So and you know, so ciprofloxacin inhibits the activity. It is inhibiting the activity of DNA gyrase because DNA, it knows DNA gyrase is supposed to do this role. So if it inhibits the activity of that, then what is going to happen? Gyrase is not going to go there and do the job, right? Understood. If gyrase is not going to do the job, then there is stress over there already. It will start breaking. So that is the first step. Second one is it also targets topoisomerase 4. Topoisomerase 4 is encoded by the PARC gene. Okay, it is not written over here. It is PARC gene. So PARC is for partitioning. When the two DNA strands, they are separating like that. When they are separating, there is again stress over there for separation. So that is the enzyme, PARC gene, or the partitioning enzyme that is going to relieve that and help them separate out. So it will target that also. So if it is doing that, responsible for separation of two interlinked daughter strands, it will prevent the DNA duplication to occur. So that's it. You understood what ciprofloxacin does. Then colistin uh, is from the polymyxin category. So that is bactericidal. Colistin was the antibiotic that was, I'm not sure which year it was discovered, but I definitely know that it was banned many years ago in 1960s, especially because it has got a lot of like side effects, internal bleeding and other things are there. So it was banned during that time. Excuse me. So it binds to the LPS, lipopolysaccharide, and phospholipids in the outer cell membrane of gram-negative bacteria. I told you yesterday, gram-positives have what? Peptidoglycan, and gram-negative have got lipopolysaccharide. Yes. So it binds to lipopolysaccharide. Okay. So lipolipid, poly, many, saccharide, sugar, like that. Some words you can know, but no one will think that much to understand. Okay, it is. But yeah, if you follow at the very basic level, then you will understand what it is. So yes, the lipopolysaccharide and phospholipids are there um, in the outer cell membrane of gram-negative bacteria. So it competitively displaces the divalent cations, calcium and magnesium, from the phosphate groups of membrane lipids. So that leads to disruption of the outer cell membrane. And when that cell membrane is disrupted, the intracellular contents that are there, they leak outside the cell. And when the contents leak outside the cell has to die because of the osmotic balance is not maintained. Okay, so that is the mode of action of colistin, which is a bactericidal agent. So now we move on to antimicrobial resistance, right? So everyone knows mostly what is AMR. So AMR is the ability of microorganism to persist or grow in the presence of drugs designed to inhibit or kill them. So that is in a very basic form, or that is the basic definition of antimicrobial resistance, is the ability of microorganisms to persist. Persist meaning they are still there, or grow in the presence of drugs designed to inhibit or kill them. So drugs are supposed to inhibit them, so lower their numbers or destroy them completely. There are some organisms that also occur in, for example, what is known as viable but non-culturable forms. You can have a sample sometimes where the organisms will be there, but you can't culture them, right? So that is another phenomenon that is there. It is not linked with AMR as such, but that is another factor that is obviously can be taken into consideration, because we as human beings, we see what we see in front of us, right? Whether it is a molecular test or whether it is an assay that we have performed or whatever. but the whole world knows. I mean, probably you will also know there are only 1% of organisms on Earth that you can grow from the universe actually. Only 1%. 1% is very small number. It is 1 out of 100, right? That you can actually grow on the agar medium. So these forms are also there. So these forms again are important. Um, and there are different ways. Some organisms, they will form spores. Spores can spread. Again, spores are the forms that allow the organisms to survive extreme conditions, desiccation and extreme heat, extreme cold, and all the other things. But not just spores, there are sometimes other forms as well that exist where you will not see that organisms are there over there. But as soon as the conditions become favorable or something changes or nutrients become available, they will come back to life, kind of. And another important thing to consider also is, you know, uh, Antarctica, right? You know what is Antarctica, right? The glaciers and all those things. So over there now, new exotic organisms are coming back to life, is what they say, because when the glaciers are melting, so the forms that people didn't see previously or that were frozen in time, whatever million years ago, they are all coming out. Um, so we need to understand, or we need to, I mean, we are looking at the definition from a very small sense, but it has a very broad meaning where, anything can happen. Cross resistance occurs, so many things are there. I'm not going in the details of all the different things that are occurring, but we just need to appreciate that some antibiotics can cross select for different traits. So factors responsible for AMR, I think I don't need to tell you what factors will select for AMR. Most common is overuse of antibiotics in humans as well as agriculture, inappropriate antimicrobial stewardship. So whether that is wrong choice of drugs, so you know you are giving the incorrect drugs, or you are giving inadequate dosing. So instead of five, you are giving only two tablets, for example, or poor adherence to treatment guidelines, um, for example, or you are giving more amount of antibiotics than needed. So it can, and as I said to you earlier, it can happen in humans as well as in the agricultural setting, in the environmental setting as well. And then, of course, we know hand hygiene, sanitation, clean water, air, all these are also very, very important because they play an important role um, in our ecosystem. So we all know that AMR is a kind of a silent epidemic or a silent pandemic. So currently, more than 35,000 people die in Europe as a result of that. I don't know in India what the estimate is like, but over here, in 2022, the Nagavi report highlights 1.2 million deaths are occurring each year due to the AMR, and then another, I think, 5 million deaths are also indirectly attributed to AMR. And previously, the Jim O'Neill report highlighted, it said that by 2050, 10 million people will die, is what they said. Now, new reports are saying 39 million people will die. Um, by 2050 as a result of that. So it is a huge number. Okay. Um, and you can see in that picture as well that AMR will kill more people than cancer and diabetes combined by 2050. So in the picture itself, you can see how the resistance is developing. You are having, the poultry, for example, or livestock you have, and you are giving antibiotics. So 80% of the antibiotics given over there, they are not necessary actually, but they are, in the past I gave you some examples of antibiotics that were used as growth promoters, like streptomycin, for example, tetracycline. So so that is one way. Then again, like fertilizing with antibiotic resistant manure. So again, in the ecosystem, uh, it is translated in the crops as well. And then you also have the consumption by humans as well, whether it is the plants or whether it is um, the poultry, for example. And another important thing is 50% of antibiotics again in humans are unnecessary. They are not needed. Another thing to also know is that plants, right? Some plants, they have the ability to actually uptake microorganisms as well, if you believe it or not. Maybe you didn't hear that before, but we in the UK have performed so many experiments, and there are certain pathotypes of E. coli, like E. coli O15. I have got a lot of press releases. You type my name and you type whatever, I don't know, any UK or uh, international newspaper, you will see all sorts of news um, related to different sorts of emerging infectious diseases that I write on, and then foodborne infectious diseases as well. So I remember one case was there when the journalist had asked me to write a report highlighting about this, the presence of E. coli O157, which is one of the pathotypes of E. coli, and it was linked to a salad leaves in sandwiches over there. So there are organisms that can go from the soil in the plant as well, and it can affect, because maybe over here in India, I don't know, we are not having that kind of every country or every nation has a different issue, right? It's not everything is same everywhere. Yes, there are some things that will be similar, but then other than that, you will have specific things that are there. So in ours, on our side in the UK, foodborne diseases and all is a very major problem over there. And if you suspect an outbreak associated with that, and if you're having sandwiches that are distributed and they are going everywhere, then you can imagine. Um, and some of these uh, diseases are not very what uh, very easy to treat. They need specialized treatment. Not all microorganisms can be given antibiotics and you can work. It is not like that. Okay, we'll learn about that as we are discussing the different lectures we've done about. So have an appreciation for that. Don't just think that, oh, through the meat or other places you can have this transmission. Don't, you can have transmission from anywhere. Even if you have the salad, if you're not washing it properly, you can see over here fertilizing with antibiotic resistant bacteria. If something is there of that, and you consume, your gut is becoming enriched in those microorganisms. That's so mean. So One Health initiative, I think everyone is maybe familiar with this term, but One Health is what? It is One Health for everyone basically. So it's a term used to describe principle which recognizes human and animal health are interconnected, so they have connection with each other. That diseases are transmitted from humans to animals and vice versa, and must therefore be tackled from different angles. So this term is globally recognized, having been widely used in EU, and in 2016 the United Nations political declaration on AMR. And you can see nicely in the picture here what the human health, animal health, and environmental health that is interconnected. That's true.
6 out of 10 diseases that are there, and 3 out of uh, 5, if I'm not wrong, new emerging diseases, they have their direct link in animals. Okay, that is another thing to remember. So, is this a new concept? And on you are learning about this now, or we are giving it a different name, like 1 year. But this man over here, anyone knows who is that man? Philosopher. He is the Greek physician and philosopher. What is his name? It's interesting. Anyone knows his name? All medical students take the oath, take his oath. So this no, Hippocrates. As famous there. So it extends to ancient times, 300, 400 BC. So Hippocrates has a very famous writing, which is basically on hairs, waters, and places. So the treatise explores how environmental factors, air, climate, water quality, and geography cases affect human health and diseases. How do they play a role um, in that one health? So obviously, planetary health is there. So he argues that to understand and treat illnesses effectively, physician must consider climate and season of a region, quality of water people drink, terrains and winds of the area, the customs and lifestyles of the inhabitants. So all different factors are there, which I'm sure you agree to all these things. Um, Yes, good thing. So these are the some of the key themes that he was highlighting. Okay, so these were some of the key themes he was highlighting, and what he highlighted is not all correct, because we already know that what we are doing over time, we are improvising and we are looking at, okay, what things are working and what things are actually true, right? Because that is science. Yesterday was the director of the institute highlighted to using some version of your Dhyan into Vidya. Like, for example, knowledge into science, conversion of knowledge to science. So there are key themes over here. One is environmental determinism, and another one is epidemiology and public health, and another one is medical geography, and the last one is ethnographic observation. So first one, he's believed that physical environment shapes not only health but also character and behavior. People from cold climates tend to be more courageous, while those uh, from warmer climates are more intelligent but less warm. So that was one of the things he highlighted. Okay, I'm not telling you to believe in all this. As I said to you, some of the things are incorrect over them. You are the ones who are going to tell. You are going to make the Vagnani, you are going to be the scientist to highlight to me which one you think is the correct, which one you think is the incorrect. The second one is epidemiology and public health. So, text is an early attempt at understanding disease patterns in population. We all know that if you have a strain spreading in a community, then you need to monitor how it's spread, where it's spread, and you need to make sure you have measures to combat that.
It emphasizes importance of observing local conditions to predict and prevent outbreaks. Okay. And third one is medical geography. It is one of the earliest works to link geography with medicine. So laying the groundwork for later fields like medical anthropology and epidemiology. Okay. And then the last one is ethnographic observations, where Hippocrates also discusses different people and cultures, often in comparative and sometimes speculative manner, reflecting the Greek worldview of the time. So what he's saying is that he is looking from his lens, from the Greek lens, right? We are, for example, our physical structure and the structure of the people elsewhere is not always the same, right? We are different, we have evolved in a different manner, but he is seeing everything from a very Greek view. Okay, that's why he's saying that he's always like comparing and speculating that okay, maybe over here this thing or that thing. So which one do you think, or which of these do you think are incorrect? Who is going to courageously tell me? You have got one, two, three, four. You raise your hand and you tell me who wants to answer.
Because we cannot decide who is courageous or who is not,
Okay.
according to the climate
How
change.
many of you agree with her? You raise your hand. One, two, three, four, five, six, seven, eight, nine. Okay, nine people agree with her. And which other one? Anything else that is coming out to you? So nine people raise their hands for environmental determinism. Gitanjali, which one do you think is incorrect? What else is incorrect over here? Okay, how many agree with her for ethnographic observation? Yeah. So how many agree with her for that one? One, two, three, four, five, six, seven, eight, nine. Okay, nine people agreed in the previous one and nine people agreed in the later one. So good, well done. You both are, whoever agreed, you are right. So these two are incorrect. Okay. So here you have got the aspect where, environment affects health, and then the definition is there: climate, geography, surrounding, influence physical well-being. Yes, Hippocrates' view is believed to directly cause disease and shape bodily humors. Scientific view is accepted, studied through environmental microbiology, epidemiology, and public health. Okay, second one is environment determines character, is wrong, incorrect. You know that climate and geography shapes personality traits, no. Moral qualities, no. Okay, so it is rejected. Personality is shaped by complex genetic, psychological, and social factors. The third one is cultural generalizations. Okay, again, these are very broad, stereotypical claims on like entire group based on limited observations. Again, this is rejected. It is biased, unethical, and scientifically invalid. Empirical observation, systematic collection of data through direct experience and measurement. Scientific view is yes, it's required, it's central to scientific methods and evidence based medicine. So, based on the data, you will be able to highlight what is true and what's false. Ethical ethnography, uh, respectful and systematic study of cultures with informed consent and cultural sensitivity. That is ethical ethnography. So it's essential, so guided by ethical standards and cultural respect, because it will tell you many things in a community, for example, if you want to solve problems in those communities. Okay, it's not like we study community for just fun, right? We are trying to understand what is happening over there, what you can do, how you can improve the health and well-being of those. people, for example. Disease mapping and prediction, again linked to epidemiology. So you have got the GIS, statistical modeling, real-time data. And then lastly, social determinants of health, the conditions in which people live, work, and age that affects health outcomes. Obviously, we know, health and other things will have an effect on the outcomes that are there. So that is again, you know, central. It includes income, education, access to care, and discrimination. So, for example, if the people are from a high income background, you have access to good health care, for example. So those kind of interrelation and correlation, social determinants of of health and well-being, and that is true in different settings as well. Whether you work in a scientific area as well, it is the same concept. When you guys are choosing, for example, universities as well, you are looking, which university is ranking better, what kind of research is going on, what facilities are there. Is a similar situation over here. So this is related to health though. So why One Health matters? So three changes: population and growth expansion. More people are living near animals, because sometimes we see that as the population is increasing, you can see in the previous days people didn't live that much on the rural side and other things. But pretty sure now everyone knows in major cities, if there are any suburban areas or whatever, they are getting filled very quickly, right? You will see the property prices and all will be increasing in those areas as the years have passed. In when you were born that time, maybe they didn't have any value, but nowadays, because there is no place for people to stay, so what is happening, that access is becoming more closer. So then zoonotic disease transmission also can be a factor over there that can uh, play a role, highlighting the spread between animals and humans. Excuse me. Then you have got the environmental and farming changes. So climate change is one of the things. Deforestation, right? When you're again moving in the rural area, if there is any trees, you are creating buildings, for example, for housing people and other things. So that also has an impact, no, on the environment, for example. So that is there. Then intensive livestock farming that disrupts sometimes ecosystems, making it easier for disease to jump from animals to humans. So if you are very much in a crowded environment, then that risk of transmission is also very high, right? It is no, I don't have to tell you. It is a natural common sense. If you are there in a crowded area, the rate at which you can get infection, if there is infectious disease in that particular hotspot is there, then the rate at which you will get is very fast, because you are at a such close proximity with the person. And you saw that, for example, in the corona time, what was happening when people were there next to each other or nearby each other, they were talking, doing something. Got is so that again needs to be considered. Global movement is another important thing. In the earlier days, people didn't use planes to travel. No, everyone was using trains and other things. But now global access is huge, so increased travel is there, and trade is there. So that allows disease to spread quickly across borders. And sometimes we have diseases, coming from like different places, in, for example, in different countries where it is not known, like it is never seen, and then suddenly it spreads. Sometimes we have, like, for example, in the UK, from Africa, we have sometimes like people who are coming with monkeypox. Monkeypox is a virus. I have written another, I have got lots of press releases. You just need to type my name and type whichever one you want, COVID or monkeypox or whatever I told you, E. coli O15, you will see all the things. They tell me then, okay, which one is more severe, which one is less severe, how is the transmission and all that. So that is also important, so understanding. And I think we all know during the COVID time that, all these borders were closed. People were telling, to do the LFTs, you know, um, the test to identify whether you have the disease or not, because the risk of transmission is very high. And that increases the burden. No, it is not a burden just on the person. It is burden on the medical facilities, burden on the government, on everyone is burdened as a result of that. So, so this is why One Health matters. So One Health, as I said, is that broad concept where humans, animal, and plant, all this, the health of that is combined together as one. So this is again just a very snapshot of everything, highlighting, social determinants and other factors in environment. For example, you can see climate change, water pollution, air pollution, deforestation, loss of biodiversity, you know, and you will be so surprised to know, I think I saw the other day in India there is uh, there was a activity that was done where they were planting mangrove trees, especially inside on the Gujarat side, for example. I know about mangrove trees because I am also from that coast of, um, Mumbai, right? So there also we have a lot of like mangrove and other things. And why those are important? Because they protect from cyclone and other disasters. Because when these disasters will strike, obviously you will have diseases, no, that are coming in and doing. So don't underestimate the health of plants and other things, because they not directly you will not think directly that, oh, plant is going to save. No, but actually it plays a very important role. And in the similar way, you have animal health also. If you are having a place where there are animals are very crowded and they are not grazing properly, they are infected with something, then that transmission will occur very quickly. For example, you don't know. We need regulation, we need policies, we need other things that can help us improve this, because only then we can make a tangible difference. So I think that is all for just now.